Today we're going to look at human variation and how that relates to Mendelian genetics or single gene traits. So on this slide, you will see that it's looking at free versus attached earlobes. And in this picture, you'll see that this earlobe is attached and this earlobe is free. So it kind of like dangles a little bit, where this one, it just hits the ear right at the bottom. Over on the right-hand side of the screen, you'll see some charts that have some information about the phenotypes of parents. So phenotypes are the physical characteristics, and if both parents had free earlobes, you'll see that 12 of their children had free offspring, and 22 of these groups of parents had 22 offspring. So this isn't like one set of parent. This is looking at multiple parents and their kids kind of combined in this chart. So it's not one family in particular. And you'll see the same for if the parents were free and attached and attached and attached. Now, what I want you to do is look at what you see in these charts and what do you observe? So what do you notice? What patterns do you notice in the relationships between parents and their offspring for the traits of earlobe shape? As you're thinking about this data, consider what steps you are taking to make sense of that data. And be sure to read underneath. It kind of gives you some information about where this data was taken. So let's think about what you learned from here. So when you were making your observations and trying to figure out what was going on, how did you attempt to make sense of this data? What strategies did you use? What did you look at in particular? And then did anything surprise you? And did anything confuse you? All right, so we're going to look at a study that Mendel did. And he was looking at a single gene dominant recessive model, and he was looking at pea plants. And in this case, in this picture, we're looking at the color of peas. So in this particular gene, the variation is either or, so it's yellow or green. The trait is controlled by a single gene. We're going to call it the Y gene. And there are two variants of the Y gene, and we call the two variants of the Y gene alleles. So they're alternate forms of genes. So again, there's two alleles of the gene in the population, capital W and lowercase w. And the trait is inherited in a dominant recessive pattern, where the dominant allele masks the recessive allele. So if you look at this, over here, little y is green. And so that tells us that little y is the gene for the version of the allele or the variant of the allele. So little y is the green allele. Capital Y is the dominant one, and it codes for yellow. So the gene or the gene for color of pea plants, again, there's two alleles or variations, yellow or green, and capital Y codes for yellow and lowercase y codes for green. So if you have two dominant capital Ys, your offspring is going to be yellow. If your offspring gets a big Y and a little y, because big Y, yellow is dominant, it's going to be yellow. And that's the same over here. And then here we have little y, green. Now, this Punnett Square is not showing that, okay, every pea plant, if this is like the seeds the egg, and this here's the pollen, the plant that determines those. What you're going to see is they're not going to always have 4 offspring. This is showing a prediction of what could occur. And so anytime these two pea plants breed, they can have 1/4 of a chance that their offspring is going to be big Y, 1/2 of a chance that they're going to be big Y, little y, and 1/4 of a chance for their offspring to be little y. So this is just a probability of what might happen. It doesn't mean that they're always going to have 4 offspring in this ratio. Just like if you flip a coin 100 times, it could be 100 times heads, it could be 100 times tails, but that's not probable. It's possible, but not probable. So what's probable if you flip a coin 100 times, it's going to be about 50-50. So this is the same thing. It's showing probability. So what you'll notice with these P's as well is when I was giving you the percentages a minute ago, it was the probability of what percent would be homozygous, dominant, 2 capital letters, or 50% are big Y, little y, heterozygous, or 1/4 is little y, homozygous, recessive. So those are probabilities of genotype. The genotype are basically the genes, or the alleles in this case. So the phenotype are the physical traits, so whether they're yellow or green. So the probability of these parents having a yellow offspring is 3/4. The probability of these parents having a green offspring, or offspring that make green peas, is going to be 1/4, 1/4. So when we look back at our images and the data that we were just observing, we need to notice, like, what are we talking about? Are we talking about genotypes, so the genes, the letters, the alleles, or were we talking about phenotype, which are the colors, when you're looking at these particular traits? All right, and how this kind of works, how you are able to make these predictions. Okay, there's some important vocab that you need to learn. Dominant, mask the effects of others. Recessive is masked if dominant is present. Dominant is always represented by a capital letter, recessive by a lower letter. Homozygous is if you have 2 of the same alleles. Heterozygous if you have 2 different alleles. And so we're going to fill out right now a Punnett Square. And so this is the egg cells from the biological egg producer. And this here are the sperm cells, which are pollen cells in plants, from the sperm donor. These pictures here are the fertilized eggs. So when the sperm fertilizes the egg, this is going to have 2 letters inside of it. All right, so if the egg has a capital Y and the sperm has a lowercase y, so dominant and recessive, then what's going to happen is the offspring is going to be capital Y, lowercase y. In this case, they're 2 different alleles. So that means that this egg is heterozygous. Now, the next egg, this one here, the egg gave a little y and the sperm donated a little y. So it's lowercase y, lowercase y. Now, if they have 2 of the same types of alleles, which in this case it does, 2 lowercase recessive alleles, it is going to be called homozygous. So this is homozygous recessive. This is heterozygous. Okay, the next one here, this capital Y was donated by the egg cell. The sperm cell donated the lowercase y. If we have 2 different alleles, that is heterozygous. Now, for our last one, this egg is going to donate a lowercase y, sperm lowercase y, and so in this case it's homozygous and it's recessive. In this image, we do not have any capital Y's, but a capital Y would be 2 of the same alleles, so homozygous, and they both are dominant if they're capital Y's. So this is how you would fill out a Punnett Square. Now, if we look at the probabilities here, it's probable that half of their offspring will be heterozygous and half of their offspring will be homozygous recessive. So if they had 100 kids over the time period, over their lifetime, about half of them are going to be heterozygous, half of them will be homozygous recessive. It doesn't mean they have to be 50-50, but it's probably going to be close. It might be like 47-53. So we can figure out kind of percentages that way and predict what the offspring might be, what's probable for them. Now, looking back at our earlobes, okay, attached and free, and thinking about how traits are inherited from the parents and the offspring, if you look at this, does this reflect what we saw with Mendelian genetics? Okay, does it fit the single gene dominant recessive model? Why do you think that? And would it be possible for two parents with the dominant phenotype to have a child of the recessive phenotype? Here's something that might help you answer those questions. Okay, so if we have two parents that are dominant, one parent's dominant, one is heterozygous, and this one, both parents are heterozygous. So we can kind of see what the predictions would be for their offspring. So if we had two homozygous dominant parents, all their kids are going to be homozygous dominant. If we had a homozygous dominant parent and a heterozygous parent, all of them are still going to have the dominant trait. Okay, if we look here with both are heterozygous, 3/4 are going to have the dominant trait, and 1/4 is going to have the recessive trait. And there's other combinations that as well. If you had two recessive parents, homozygous recessive parents, what would their offspring look like? So think about what this might mean when you're looking at these charts. Okay, and we're not able to tell whether the parents or the children are homozygous in these numbers here. But we know what their phenotype looks like. So we know what should be dominant by looking at these numbers. You can make some comparisons. Okay, would it be possible for two parents with the recessive phenotype to have a child of the dominant phenotype? All right, so which data fit the model and what if they don't? What you're going to work on is you're going to have 4 traits to look at on a handout. And you're going to make observations, create Punnett squares, test things out to answer the following questions. Which traits fit the single gene dominant recessive model? How do you know? And if they don't fit the model, what else might explain the data? So you're going to look at this handout, and you just need to do one of the 2 pages. There's an A and a B. And it has the traits, pictures of what the traits look like, and some data. And so the questions that you're going to answer are up at the top. Which traits fit the single gene dominant recessive model? How do you know? And if they don't fit the model, what else might explain that data? You'll then turn in this document in class.